I'm ready.
All right, hello everybody. I'm Trey. This is my boss, Dr. JP Clancy.
Hi, how are you? Yeah, I just want to comment. I really like the hair. I hope you do too. If you do, give him a thumbs up, okay?
JP has hyped me up for my hair. I want to thank JP first and foremost for giving me the opportunity to work in CF. This has been an awesome experience over the last 2 years, and actually my 2-year anniversary comes up in 6 days.
Has it really been 2 years?
Yeah, it's been 2
awesome.
years. Yeah.
It's been great to have Trey as part of the lab. I think you've brought a dimension that obviously didn't exist, and I don't know. It's been a tremendous benefit to us too. So
guinea pig.
Etc.
Did you hear about the NPD yesterday?
I did hear. Yes, I'm going in there on Tuesday, and
I think
it will work again.
We need you and Keith.
We need some new eyes.
Yeah, yes. We need you and Keith to figure it out. There that goes. Anyhow, I want to kind of start off and say, first off, ask what kind of drew you to CF? You didn't choose before med school. You didn't aspire to be a CF physician, correct?
No, I had no experience at all with CF. I'd never knowingly met a CF patient before I got into medical school. I worked in a lab between third and fourth year of med school, and it happened to be a lab that did CF research.
Awesome.
It happened to be a guy named Mike Welsh, who's kind of known in the field.
Yeah.
He's made the CF pig, and some of them are kind of important fundamental discoveries about CF, and he happened to be there.
I think that's how a lot of people get into CF. Mm-hmm. It's a pretty fascinating disease, and I think that even that's why I got a chemistry degree, is because I was fascinated with the biochemistry behind it. So in your words, because I've written about what we do a little bit, I kind of went, I guess how would you explain what we do in, I guess, relatively layman's terms?
Sure. So we have a pretty...
I guess like the theratyping, the personalized medicine.
Sure. I was going to say we have a pretty broad sort of interest in sort of CF research across the institution that begins at the very much the cell level and goes all the way to the sort of the quality improvement and defining best practices. So everything in between. But the area that our lab is most focused on right now is doing this, basically better understanding rare CF causing mutations and whether they have any evidence of sort of being responsive to some of the drugs that are either available or in development for CF. There's a term we use, it's called theratyping, which is sort of used in a lot of different ways, but the general idea is that you're basically trying to characterize or understand a mutation based on how it functions and using those kind of CFTR modulators as kind of almost a probe, as a way to sort of test.
I think that's really cool about. I think that's really interesting that's what we do. I mean, obviously for every reason, the model systems, everything, but I think it is cool to think that we're almost like reverse engineering how we understand the rare mutations as opposed to going at it from all the other genetic approaches you can take.
Yeah, I mean, don't get me wrong, there's plenty of reasons to continue to look for new ways to treat rare mutations, but I think there's some evidence out there that some of these rare mutations may be responsive, and some of them aren't easily studied in cells that are not derived from a patient. Some of them require actually that some of these things called splice mutations are just really hard to make in a laboratory. So the best source of those really is cells that come from a patient.
So what is the backstory of our model system? So we don't necessarily have to get into all the minutiae of the monolayers and the Using chamber and all the approaches we use, but what is the backstory behind monolayers and spheroids? Is that's not a technique that was pioneered in our lab necessarily? The spheroids are...
Yeah, the spheroids, I think, at least in part, I mean, there was two groups, at least a couple groups working on it at the same time. I think we actually officially got published slightly before. But anyway, it was close.
Yeah.
But we do go at it a little differently. So monolayers are basically when you grow cells as a sheet.
Right,
and they sort of form what we call an epithelium, which is where they have sort of a nice tight connection. The cells are all connected and working as a team, if that makes sense. And we can measure how salt moves across those. And
that matters because CF is a membrane protein.
And salt is
CFTR.
We believe salt's the problem.
Yes.
You can argue which salt, sodium chloride bicarbonate, clearly salt. And water. And so the Using chamber is a device that allows you to study how the ions are moving across the layer of cells. That's been around since the 40s.
Yeah.
And so
with our success rate with the actual device working, it seems
Lately,
like
it's been misbehaving, but it will rise again. But what was, I guess, historically people have looked at cells that are derived from patients who've undergone transplant. And we've taken lung cells where you get lots and lots of cells, and you can seed them onto these little growth membranes, they're called, and then you grow them up, let them mature, and then you can go and study them. And that's great, and we will continue to do that. However, if you want to study cells from a patient, that's not very feasible. So we had to come up with some ways to adapt some of the newer understandings of how to go from a few cells to enough cells to be able to do a study.
Okay, so I didn't realize that you and so was it mainly Alicia and you, like the lab group here that pioneered?
There were a number of labs doing it at about the same time, but in 2013 there was a report of a group who had found the cells that normally line your nasal membrane or in your lungs, in your airways, they're fully differentiated, meaning they're sort of done. They've kind of achieved being a cell. They're mature. They don't divide anymore.
Oh, so when you brush them, it's hard to.
Yes, those cells that come off mostly are sort of fully differentiated. They don't like to divide. Maybe you can get one or two, but some people came up with some really ingenious ways of taking these differentiated cells and making them take a step back in differentiation so that they would divide a little bit and expand. You get to go from maybe 100,000 cells to a couple million. Okay. And that really allowed us to go from a brush to a monolayer.
Gotcha.
And so that was a big breakthrough. But there was a lot of questions like, how do you grow them? Do they, which is the best media? How many, all sorts of.
Which is all the variables of science.
And we finally figured that out. And sort of hand in hand with that, we started to study. There's a lot of work that happens at this institution looking at organoids, which for different reasons. I mean, a lot of it's related to understanding how to, actually regrow intestines. Mm-hmm. But we adapted some of those techniques with the nasal cells, and they grow up a little faster that way. And what it gives us, we can basically study the nasal cells two ways at the same time. One to look at the salt transport, and the other really look at the fluid transport.
Right, and I think that's really cool. And so, as I've written, that's mainly what I do is the data analysis of those after the cells have been differentiated and applied. The oozing chamber and the monolayer or spheroids have been looked at. I actually do most of the data analysis. Yeah. So I've written about that, and I think that that's really cool that I get to see the actual data of cells. Safety are improving.
I'm very spoiled because I get the product of the data that you've looked at.
The data, it can be cumbersome, but it is. I enjoy it.
It's one could argue a little tedious, but
well, that's okay.
No, it's fun. When something really works, it's pretty. Pretty rewarding when you see that.
So I guess I'm going to ask you a little bit about. I want to be careful to prognosticate because CF medicine's it's tough, but
it's also changing so fast.
Yeah.
It's really hard.
What do you believe is the future of the theratyping, like what we do specifically? Do you think that essentially modulators are going to be so good that you won't even have to look at patients anymore because it's going to be cover 100%? Because I know the triple combo therapy is hoping it's in phase 2.
It's actually in phase 3.
It's in phase 3.
Yeah.
The hope is that it's going to cover 95% of mutations.
Roughly.
So I guess. What is the euphemism in science that you spend 90% of the time spending trying to advance the last 10%? It's really any task you spend. Yeah. But I guess what happens for those patients? Do you think that there eventually will be modulators that cover everything? It's a good question.
Things are changing pretty fast, and that's great. I would say that by and large, most people who have a delta 508 mutation, or at a minimum one of these other sort of clearly ivacaftor responsive medications, those ones theoretically will have access to what I would consider should have access to highly effective therapy, assuming these triple combo studies are successful. So what's the role of theratyping? I think the short-term role is there are those patients out there who don't fall into that big bucket, and are any of these available or modulators in development make sense for them. And I think so there's going to be a small group of patients who, and within that group there's maybe a smaller group who actually we can demonstrate some activity. Now again, does it guarantee access? It's still something that we're working on. Yeah. We're trying to be able to give the information in a very, the results in a very in a way that would be supportive for conversations with third-party payers, meaning insurance companies. Yeah. we'd love to get to a point where the data is strong enough that it actually could be used for regulatory purposes, which means the FDA approvals.
So,
what does that take?
Yeah, because I know that's kind of what we're working on.
Yeah,
right now,
it's not easy. I mean, you have to basically have a test that is incredibly reliable. You feel extremely confident that what happens in the test tube, basically, or in the cells, is predicting what's happening in the patient.
So in vivo and in vitro, which becomes difficult to measure, especially with sweat chloride. I mean, you just don't know what. Well, trying to prove, trying to corroborate the experiments we do with the biomarkers that patient actually responds.
So one way we can get at it, sort of using what's available now, is seeing how well the nasal cell responses predict a variety of different clinical responses, be it sweat chloride, lung function, risk of exacerbation, growth, all those things. Maybe come up even with a panel of things that we could say this quote equals a response.
Right, yeah.
And it might have to depend on where you are in your disease. Some people who have very mild disease, lung function doesn't mean much because it doesn't get better.
Exactly, that's kind of, I think, what was my hope when I officially got on.
Yeah. So I think so. There's definitely going to be that role, and you can see how the bar for to get an insurance company may be a lot easier than getting an FDA approval. I won't say it's not impossible, but it's going to take more work.
Yeah.
The other thing is, I think that I think we all want to be in a world where there's more choices than just.
That's what I was going to say. You're still going to need theratyping to figure out what the best modulator is.
Yeah, and I think
patients.
If we've got a few choices, I think being able to give data that is truly predictive. Now, that still requires that first thing, is validating that these tests are really predictive of a clinical benefit. Because all of a sudden, if let's say my drugs or my nose testing says, oh, you should be on Orkambi and not Symdeko.
Right.
Well, you better be pretty confident. Cause that's a lifelong decision, possibly.
Of course. So, and then at what point do you evaluate the patient and say, well, the Orkambi's not working as well as we'd hoped, so let's switch to Symdeko, and then you just get into weird effects when you don't know the full mechanisms of what you're doing. And
so there's some kind of a short term where you might be able to get people access for trials and look to see, there may be a home run. I mean, if you have a big, your sweat drops from 100 to 25 and your lung function pops up 10 points, it's easy. You clearly you've benefited. It's more where it's subtle. You drop 15 points in your sweat and your lung function went up 4 points, but your weight's not. That's where it gets tricky. And that's what we're going to wrestle with.
Yeah. Well, I'll go ahead and close this down just to give you time, but I want to thank you, JP. I'm really excited for what we do in the lab, and I'm looking forward to the future. I think CF medicine is bright right now. Science is bright. Dr. JP Clancy is a pioneer in CF medicine, and we're thankful for everything he does. So
keep it coming. All right. Did you show him our really fancy lab?
I didn't. I can give you guys a tour.
You guys should take a tour.
I can give you a tour.
It's underwhelming, I promise.